gut tube and body's cavity. A tube on top of a tube. During the 3rd and 4th weeks, the top layer, ectoderm, of the trilaminar embryonic disc forms the neural plate that rolls up into a tube to form the brain and spinal cord by the process called neunüation. Almost simultaneously, the ventral layer, endoderm, rolls down to form the gut tube, such that the embryo consists of a tube on top of a tube, the neural tube dorsally and the gut tube ventrally, Fig.1. The middle layer, mesoderm, holds the two tubes together, and the lateral plate component of this mesoderm layer also splits into visceral, splanchnic, and parietal, somatic, layers. The visceral layer rolls ventrally and is intimately connected to the gut tube. The parietal layer, together with the overlying ectoderm, forms the lateral body wall folds, one on each side of the embryo, which move ventrally and meet in the midline to close the ventral body wall, Fig. 1. The space between visceral and parietal layers of lateral plate mesoderm is the primitive body cavity, which at this early stage is a continuous cavity, because it has not yet been subdivided into the pericardial, pleural, and abdominopelvic regions. Formation of the body cavity. At the end of the 3rd week, intraembryonic mesoderm differentiates into one paraxial mesoderm, which forms somatomers and somates that play a major role in forming the skull and vertebrae. 2 intermediate mesoderm, which contributes to the urogenital system. 3 and lateral plate mesoderm, which is involved in forming the body cavity. Soon after it forms as a solid mesodermal layer, clefts appear in the lateral plate mesoderm that coalesces to split the solid layer into 2. 1. The parietal, somatic, layer adjacent to the surface ectoderm and continuous with the extraembryonic parietal mesoderm layer over the amnion. Together, the parietal, somatic, layer of lateral plate mesoderm and overlying ectoderm are called the somatopleural. 2. The visceral, splanchnic, layer adjacent to endoderm forming the gut tube and continuous with the visceral layer of extraembryonic mesoderm covering the yolk sac, Figs.ib. Together, the visceral, splanchnic, layer of lateral plate mesoderm and underlying endoderm are called the splanchnopleural. The space created between the two layers of lateral plate mesoderm constitutes the primitive body cavity. During the 4th week, the sides of the embryo begin to grow ventrally forming 2 lateral body wall folds, Fig.ib and c. These folds consist of the parietal layer of lateral plate mesoderm, overlying ectoderm, and cells from adjacent somates that migrate into the mesoderm layer across the lateral somatic frontier. As these folds progress, the endoderm layer also folds ventrally and closes to form the gut tube, Fig. id and e. By the end of the 4th week, the lateral body wall folds meet in the midline and fuse to close the ventral body wall, Fig. 1c. This closure is aided by growth of the head and tail regions, folds, that cause the embryo to curve into the fetal position, Fig. 7.2. Closure of the ventral body wall is complete except in the region of the connecting stalk, future umbilical cord. Similarly, closure of the gut tube is complete except for a connection from the midgut region to the yolk sac called the vitelline, yolk sac, duct, Fig.2d. This duct is incorporated into the umbilical cord, becomes very narrow, and degenerates with the yolk sac between the 2nd and 3rd months of gestation. Note that throughout the process of body cavity and gut tube development, the parietal and visceral layers of lateral plate mesoderm are continuous with each other at the junction of the gut tube with the posterior body wall Fig.we, serous membranes. Some cells of the parietal layer of lateral plate mesoderm lining the body wall of the primitive embryonic cavity become mesothelial and form the parietal layer of the serous membranes lining the outside of the peritoneal, pleural, and pericardial cavities. In a similar manner, some cells of the visceral layer of lateral plate mesoderm form the visceral layer of the serous membranes covering the abdominal organs, lungs, and heart, Fig. 1e. Visceral and parietal layers are continuous with each other as the dorsal mesentery, which suspends the gut tube from the posterior body wall into the peritoneal cavity. Dorsal mesentery extends continuously from the caudal limit of the foregut to the end of the hindgut. Ventral mesentery exists only from the caudal foregut to the upper portion of the duodenum and results from thinning of mesoderm of the septum transversum, a block of mesoderm that forms connective tissue in the liver and the central tendon of the diaphragm, Figs. 2d and 5. These mesenteries are double layers of peritoneum that provide a pathway for blood vessels, nerves, and lymphatics to the organs. Clinical correlates: ventral body wall defects. Ventral body wall defects occur in 1. The thorax, 2. Abdomen, 3. And pelvis and involve 1. The heart, ectopia cordis, 2. Abdominal viscera, gastroschisis, 4. Urogenital organs, bladder or cloacal exstrophy, depending upon the location and size of the abnormality. The malformations are due to a failure of the ventral body wall to close and probably involve the lateral body wall folds to a greater extent than the head and tail folds. Thus, one or both of the lateral body wall folds fail to progress ventrally, or there are abnormalities in the fusion process once they meet in the midline. An omphalocele also represents a ventral body wall defect. However, its primary cause is not due to inhibition of body wall closure. Instead, this abnormality occurs when a portion of the gut tube fails to return to the abdominal cavity following its normal herniation into the umbilical cord. Ectopia cordis occurs when lateral body wall folds fail to close the midline in the thoracic region, causing the heart to lie outside the body cavity, Fig. 3a. Sometimes, the closure defect begins at the caudal end of the sternum and extends into the upper abdomen, resulting in a spectrum of abnormalities called ventral pentalogy. This spectrum includes 1. Ectopia cordis, 2. Defects in the anterior region of the diaphragm, 3. Absence of the pericardium, 4. Defects in the sternum, 5. Abdominal wall defects including omphalocele and gastroschisis. Note: omphaloceles that may occur in ventral pentalogy are secondary to the body wall closure defect, not primary. The closure defect reduces the size of the abdominal cavity and prevents the return of the intestinal loops from the umbilical cord. Gastroschisis occurs when body wall closure fails in the abdominal region, Fig. 3s. As a result, intestinal loops herniate into the amniotic cavity through the defect, which usually lies to the right of the umbilicus. The incidence of gastroschisis is increasing 3.5/10,000, and it is most common in infants from thin women younger than 20 years of age. The defect can be detected by fetal ultrasound and by elevated afetoprotein AFP concentrations in maternal serum and the amniotic fluid. The malformation is not associated with chromosome abnormalities, but other defects occur in 15% of cases. Affected loops of bowel may be damaged by exposure to amniotic fluid, which has a corrosive effect, or by twisting around each other volvulus and compromising their blood supply. Bladder or cloacal exstrophy results from abnormal body wall closure in the pelvic region. Bladder exstrophy represents a less severe closure defect in this region and only the bladder is exposed, Fig. 7.3c. In males, the penis may be involved and epispadias be in the dorsum of the penis is common. Cloacal exstrophy results from AM or severe failure of body wall closure in the pelvis such that the bladder and rectum, which are derived from the cloaca, are exposed, Fig. 3d. Omphalocele represents another ventral body wall defect, Fig. 4, but it does not arise from a failure in body wall closure. Instead, it originates when portions of the Gutti tube, the midgut, that normally herniates into the umbilical cord during the 6th to the 10th weeks, physiological umbilical herniation, fails to return to the abdominal cavity. Subsequently, loops of bowel, and other viscera, including the liver, may herniate into the defect. Because the umbilical cord is covered by a reflection of the amnion, the defect is covered by this epithelial layer. In contrast, loops of bowel in gastroschisis are not covered by amnion because they herniate through the abdominal wall directly into the amniotic cavity. Omphalocele, which occurs in 2.5/10,000 births, is associated with high mortality rates and severe malformations, including cardiac abnormalities and neural tube defects. Furthermore, Chromosome abnormalities are present in 15% of cases. Like gastroschisis, omphalocele are associated with elevated AFP concentrations. Diaphragm and thoracic cavity. The septum transversum is a thick plate of mesodermal tissue occupying the space between the thoracic cavity and the stalk of the yolk sac, Fig. 0.5 ab. The septum is derived from visceral, splanchnic, mesoderm surrounding the heart and assumes its position between the primitive thoracic and abdominal cavities when the cranial end of the embryo grows and curves into the fetal position, Fig. 2 bd. This septum not separate the thoracic and abdominal cavities completely but leaves large openings, the pericardial canals, on each side of the foregut, Fig. 5b. When lung buds begin to grow, they expand caudalaterally within the pericardial canals, Fig. 0.5c. As a result of the rapid growth of the lungs, the pericardial canals become too small, and the lungs begin to expand into the mesenchyme of the body wall dorsally, laterally, and ventrally. Ventral and lateral expansion is posterior to the pleuropericardial folds. At first, these folds appear as small ridges projecting into the primitive undivided thoracic cavity. With expansion of the lungs, mesoderm of the body wall forms two components, Fig. 0.6, 1, the definitive wall of the thorax. 2. The pleuropericardial membranes, which are extensions of the pleuropericardial folds that contain the common cardinal veins and phrenic nerves. Subsequently, descent of the heart and positional changes of the sinus venosus shift the common cardinal veins toward the midline, and the pleuropericardial membranes are drawn out in mesentery-like fashion, Fig. 6a. Finally, they fuse with each other and with the root of the lungs, and the thoracic cavity is divided into the definitive pericardial cavity and two pleural cavities, Fig. 65. In the adult, the pleuropericardial membranes form the fibrous pericardium. Formation of the diaphragm. Although the pleural cavities are separate from the pericardial cavity, they remain in open communication with the abdominal, peritoneal, cavity by way of the pericardial canals, Fig. 5b. During further development, the opening between the prospective pleural and peritoneal cavities is closed by crescent-shaped folds, the pleuroperitoneal folds, which project into the caudal end of the pericardial canals, Fig. 0.7a. Gradually, the folds extend medially and ventrally, so that by the 7th week, they fuse with the mesentery of the esophagus and with the septum transversum, Fig. 7. Hence, the connection between the pleural and peritoneal portions of the body cavity is closed by the pleuroperitoneal membranes. Further expansion of the pleural cavities relative to mesenchyme of the body wall adds a peripheral rim to the pleuroperitoneal membranes, Fig. 7c. Once this rim is established, MYOB lasts originating from somates at cervical segments 3 to 5, C3-5, penetrate the membranes to form the muscular part of the diaphragm. Clinical correlates. Diaphragmatic hernia is a congenital diaphragmatic hernia, one of the more common malformations in the newborn 1/2,000, is most frequently caused by failure of one or both of the pleuroperitoneal membranes to close the pericardial canals, Fig. 0.8. In that case, the peritoneal B and pleural cavities are continuous with one another along the posterior body wall. This hernia allows abdominal viscera to enter the pleural cavity. In 85% to 90% of cases, the hernia is on the left side, and intestinal loops, stomach, spleen, and part of the liver may enter the thoracic cavity. The abdominal viscera in the chest push the heart anteriorly and compress the lungs, which are commonly hypoplastic. A large defect is associated with a high rate of mortality, 75% from pulmonary hypoplasia and dysfunction. Occasionally, a small part of the muscular fibers of the diaphragm fails to develop, and a hernia may remain undiscovered until the child is several years old. Such a defect, frequently seen in the anterior portion of the diaphragm, is a parasternal hernia. A small peritoneal sac containing intestinal loops may enter the chest between the sternal and costal portions of the diaphragm, Fig. 0.8. Another type of diaphragmatic hernia, esophageal hernia, is thought to be due to congenital shortness of the esophagus. Upper portions of the stomach are retained in the thorax, and the stomach is constricted at the level of the diaphragm. Thus, the diaphragm is derived from the following structures: the septum transversum, which forms the central tendon of the diaphragm. The two pleuroperitoneal membranes. Muscular components from somates at cervical segments 3 to 5. The mesentery of the esophagus, in which the crura of the diaphragm develop, Fig. 7c, during the 4th week. The septum transversum lies opposite cervical somates, and nerve components of the 3rd, 4th, and 5th cervical segments of the spinal cord grow into the septum. At first, the nerves, known as phrenic nerves, pass into the septum through the pleuropericardial folds. This explains why further expansion of the lungs and descent of the septum shift the phrenic nerves that innervate the diaphragm into the fibrous pericardium, Fig. 6. Although the septum transversum lies opposite cervical segments during the 4th week, by the 6th week, the developing diaphragm is at the level of thoracic somates. The repositioning of the diaphragm is caused by rapid growth of the dorsal part of the embryo, vertebral column, compared with that of the ventral part. By the beginning of the 3rd month, some of the dorsal bands of the diaphragm originate at the level of the first lumbar vertebra. The phrenic nerves supply the diaphragm with its motor and sensory innervation. Because the most peripheral part of the diaphragm is derived from mesenchyme of the thoracic wall, it is generally accepted that some of the lower intercostal, thoracic, nerves contribute sensory fibers to the peripheral part of the diaphragm. Summary: at the end of the 3rd week, the neural tube is elevating and closing dorsally while the gut tube is rolling and closing ventrally to create a tube on top of a tube. Mesoderm holds the tubes together and the lateral P8 mesoderm splits to form a visceral, splanchnic, layer associated with the gut and a parietal, somatic, layer that, together with overlying ectoderm, forms the lateral body wall folds. The space between the visceral and parietal layers of lateral plate mesoderm is the primitive body cavity. When the lateral body wall folds move ventrally and fuse in the midline, the body cavity is closed, except in the region of the connecting stalk. Here the gut tube maintains an attachment to the yolk sac as the yolk sac, vitelline, duct. The lateral body wall folds also pull the amnion with them so that the amnion surrounds the embryo and extends over the connecting stalk, which becomes the umbilical cord. Failure of the ventral body wall to close results in ventral body wall defects, such as ectopia cordis, gastroschisis, and exstrophy of the bladder and cloaca. Parietal mesoderm will form the parietal layer of serous membranes lining the outside walls of the peritoneal, pleural, and pericardial cavities. The visceral layer will form the visceral layer of the serous membranes covering the lungs, heart, and abdominal organs. These layers are continuous at the root of each organ as the organs lie in their respective cavities. This relationship is similar to the picture created when you stick a finger organ into the side of a balloon, the layer of the balloon surrounding the finger organ being the visceral layer, and the rest of the balloon being the somatic or parietal layer. The space between is the primitive body cavity. The two layers of the balloon are continuous at the base root of the finger. In the gut, the layers form the peritoneum and in places suspend the gut from the body wall as double layers of peritoneum called mesenteries. Mesenteries provide a pathway for vessels, nerves, and lymphatics to the organs. Initially, the gut tube from the caudal end of the foregut to the end of the hindgut is suspended from the dorsal body wall by dorsal mesentery. Ventral mesentery, derived from the septum transversum, exists only in the region of the terminal part of the esophagus, the stomach, and the upper portion of the duodenum. The diaphragm divides the body cavity into the thoracic and peritoneal cavities. It develops from four components: 1. Septum transversum, central tendon. 2. Pleuroperitoneal membranas. 3. Dorsal mesentery of the esophagus. 4. Muscular components from somates at cervical levels 3 to 5, C 3 to 5, of the body wall. Because the septum transversum is located initially opposite cervical segments 3 to 5 and because muscle cells for the diaphragm originate from somates at these segments, The phrenic nerve also arises from these segments of the spinal cord. C3, C4, and C5 keep the diaphragm alive. Congenital diaphragmatic hernias involving a defect of the pleuroperitoneal membrane on the left side occur frequently. The thoracic cavity is divided into the pericardial cavity and two pleural cavities for the lungs by the pleuropericardial membranes.